now I have to do my job here. That is, ASLO is paying me a lot of money to present Yves Prairie today. So I'm going to introduce him. It's a really great pleasure and honor to me to introduce Yves Prairie. Yves is a full professor at the Université du Québec at Montreal, and currently also he holds an UNESCO chair in global environmental change. Previously, he has served as president of the International Society of Limnology from 2013 to 2019, and also he has been as president of the Society of Canadian Limnologists from 1999 to 2002. His scientific trajectory started with a PhD at the University of McGill in Montreal and a postdoc at the University of Constance in Germany. Then he returned to Canada as an assistant professor at the University of Quebec in Montreal, where he is now. He has been a visiting professor at different institutions around the world, including IMEDEA in Mallorca. He has more than 100 papers, and consequently, he's very well-known limnologist. For me, besides being a good friend, is one of these current true limnologists. That is, he's putting lakes and reservoirs in the map of the global carbon cycle. For him, lakes and reservoirs belong to the terrestrial sphere. Yves is a true limnologist that goes beyond biology, including physical and biogeochemical processes in carbon dynamics. More importantly, he's teaching us that despite the valuable Lindemann contribution to limnology, a lake clearly is not an isolated microcosm. Hope you enjoyed his talk entitled CO2 and Methane in Lakes and Reservoirs, from biochemical processes to global emission. Thanks.
Well, thank you very much, Isabel, for this really kind introduction. I will try to share my screen right away. Do you see my full screen or my presentation screen?
Presentation or the notes version.
Now I presume it's better. Yes. So thank you very much again. And what I'd like to do today in this presentation is to talk about methane and carbon dioxide in both lakes and reservoir. The first half will be more about lakes, the second half more about reservoirs, and really trying to bridge the nitty-gritty of the biogeochemical processes and how that links to global emissions. But before I do that, I'd like to acknowledge the immense contribution of my past and current graduate students, postdoc, research associate on the left, and on the right there, there's Paul DeGiorgio, my long-term collaborator. I think for many of our students, we have become kind of an old couple. The only remaining unknown is who is mom and who's pops. And down from Paul is John Harrison from Washington State University, who has been a great collaborator in the reservoir bit, which we'll discuss more in the second half. So I think there is no longer, certainly not to this crowd, any questions as to the relevance of inland aquatic systems in the global carbon cycling. This is no longer into question. For lakes, I think it's very clear. This is how the amount of carbon that is processed by inland waters has gone up through time, from Cole's 2007 paper all the way to the latest assessments of that and new components of this carbon processed by inland waters. And basically we're now at 5 petagrams of carbon that is kind of processed annually in inland waters. I remind you that 5 petagrams is about the equivalent of the combined net land and ocean sinks annually. So this is a very large amount. For reservoirs, it is also quite important, and perhaps the most concrete expression of that importance is that in 2019 in Kyoto, during one of the IPCC sessions, plenary sessions, it was a new methodology report was formally adopted, and for the first time, reservoirs, which are included in wetlands, reservoirs have to be accounted for by countries that are signatories of these large international accords in their annual national inventory of greenhouse gases. In the basics of the IPCC methodology is basically that you have a bunch of tables of emission factors. They're basically CO2 and methane emission factors per unit area, and they are applied to new or old reservoirs, to methane, to carbon dioxide, and the values will depend also on where you are on the planet in terms of climate zones.
Could you
Yes?
enlarge your presentation? We are seeing your presentation really small. Could you please enlarge it?
I will try to do that. I may have to restart it, or maybe, I think I know why. So I'm going to have to restart it for a second, I think. So if I do this. No, this is not any different.
Okay. In the meantime, that Yves is trying to solve his PowerPoint problem, just to remind you that tomorrow we will have the Ramon Marguleff Award for Excellence in Education and Jens Schindler Early Career Award. And also Dr. Katarina Pank will give her plenary talk tomorrow on trace metals. And there will be workshop and associate editor meeting planned after the plenary. And you still have time to submit your entries for the fun challenge, the running challenge, the art and science corner, and the photography contest. I think now, I think it's good now, right?
It's better. Is it better?
Yes. No?
Okay, perfect.
Okay, thank you.
Yes,
Perfect.
that's for most of them.
Okay. So yeah, so basically the IPCC methodology for reservoir is a bunch of emission factors for different climate zones and age of reservoirs. But all of these things are measured, the importance is measured in terms of emission. And what I'd like to do instead in this talk, and my premise here, is that the emission is really just the net balance between the production of a particular gas and its consumption by various processes. And while it's hugely important to quantify emission, of course, in various types of ecosystems, my premise is that our ability to predict how future emissions will strongly depend on our understanding of the drivers and of production, of course, and the drivers of consumption. And more importantly, that these drivers are not necessarily the same in in the two sides of that equation. So, but to begin with, let's look at the magnitude of the emissions themselves, and then we can unpack this into production and consumption for some of the aspects of this. And the emission here to the atmosphere, this is mostly the work done by Jiyeon Kim, who's part of this Lake Pulse project. Lake Pulse project is a pan-Canadian research project that sampled about 660 lakes over a 3-year period. This is kind of a snapshot sampling type. And methane and carbon dioxide emission was measured in all of these systems. And the sampling of those lakes was kind of random according to ecozones, and also there's some human component in there. But just to look at the average rates for today, the average lake in Canada emits about 283 milligrams of carbon per meter square per day. And in terms of methane, that's about 16.6 milligrams of methane as carbon per meter square per day. Now, if you combine those things and you express this as a carbon dioxide equivalent using the GWP of 34 for 100 years time horizon, well, that gives about 1.8 tons of CO2 equivalent per kilometer square of lake surface area per day. So the question is that a lot? Well, I think it is a lot, and I hadn't, even though I have been dealing with fluxes from lake surface areas for a long time, it's only when Jiyeon came up with this comparison that I thought, this is a good way of expressing how intense the emissions are. If we take the average Canadian lake in both those two gases, well, a 1 kilometer square lake emitting at those average rate is the equivalent of driving this Hyundai Sonata for 10,000 kilometers every day. So that's quite a bit for a kind of a smallish lake by any standard. We can do also a second comparison. I come from Canada, and Canada has to report its annual anthropogenic GHG footprint in what's called the GHG inventory reports. And, you know, all sectors combined, so from transportation to aviation, well, that's part of transportation, everything, it amounts to about 700 megatons of CO2 equivalent per year. Now, just a couple of weeks ago, there was a paper that was published about natural climate solutions for Canada. By natural climate solution, what is meant here is what can we do to our systems in terms of better management of forests, of agriculture, of soils, so that we can enhance potentially the sink term of GHG in Canada. And if all of those things were to be done, all these management scenarios or these management practices, it might lead, if all goes perfectly according to plan, to a new sink of about 78.2 megatons of CO2 equivalent per year. So about 10 to 11% of the current footprint. Now, what are lakes in comparison to that? Well, if we again use the same average rates shown earlier from the Lake Pulse program and apply this to all of the Canadian lakes, and here I'm ignoring the millions of tiny lakes in the Arctic, I'm just talking about the kind of the mainland. And well, that amounts to 400 megatons of CO2 equivalent per year. These are natural emissions. So before we can, before we implement, or if we do implement those nature-based climate solution for Canada, I think it would be important to see what are lakes doing in that same time. Increasing lake emission for whatever reason, either through eutrophication or simply from climate change by 20%, well, will basically offset any mitigation that is done based on natural climate solutions. Okay, so I'm going to go to some examples of what I call the importance of understanding processes in constraining emissions and therefore being able to estimate it. So the first one will be about source and transport of methane. Then I'll talk a little bit about methane production, and I'll mostly talk about methane oxidation in this particular talk. So the current view of methane emissions from lake, and this is mostly the work of Tony Del Centro when she was a postdoc with us, is that methane originates, the methane in surface waters originate really from the littoral sediments. And the dashed line here represent a stratified lake, so this would be the thermocline. So the methane comes from the littoral sediment and moves in gradually. Well, if that's the case, that makes two strong predictions. The first is that there should be an inverse relationship between lake size and methane concentration, simply because as you increase the size, the perimeter to the surface area ratio will decrease a lot. And this is exactly what one finds, a very strong negative relationship between methane concentration and lake size that is basically derived from this idea that it comes from littoral sediments. The second prediction from that same hypothesis is that within a lake, there should be an inverse relationship between the methane concentration and the distance from shore. The idea, again, is very simple. If methane in the surface layer is produced in the margin, at the margin of lakes, well, as this methane moves towards the center, it is emitted at the surface. And by the time it gets to the middle, so to speak, well, it has already reduced its concentration. And again, we've measured this in many systems of different sizes, and indeed, this is what we find. But this effect is actually size dependent. Of course, in a very small lake, you don't have much time for this effect to be materialized. And so those two curves are kind of the profile of methane, relative profile that is from the shore to the other shore, and the middle would be the center of the lake. The two colors of those profiles are for different configurations of lakes, whether you have a round lake or you have a thin, elongated one. But what is important is that there's this big size dependent effect. In a small lake, you will still find maybe 80% in the center of what there is in the shore. Whereas in a big lake, you will find almost nothing relative to what's in the shore. And this, of course, this has huge consequences to the estimation of gas flux from lakes, because if you just go and sample a lake of, let's say, 500 meters of radius, and you just go in the center, well, typically you will be measuring half of what the spatially integrated flux would have been on the whole lake surface. So this is understanding that component is usually important to estimating correctly the flux emanating from that lake. In that same project, we looked at, we also took not only concentration samples, but isotopic samples, and we developed a little model, and I don't want to go into the details of it, but basically, in most cases, the patterns that we saw, that I showed earlier, the decreasing concentration from shore to center, could not be entirely explained by the physical processes of horizontal diffusion, of gas exchange, etc. And the isotopic analysis allowed us to look at this. and to see, well, in most cases, there was a missing source to explain what we were indeed finding in the middle of those lakes. In some cases, there was kind of a missing oxidation, so to speak. I'll get back to this later. A former PhD student looked at that in a series of 5 lakes over an ice-free season, and basically he estimated the net production of methane in the oxic zone, which is now a hotly debated topic, but he didn't measure it directly, but by difference. So instead of trying to measure it directly, what he did is he estimated all of the components of the mass balance, so all of the other components, and if all goes well, this should basically, if there's no methane production in the surface, there should be this, the sum of these terms should be 0. And to go over this quickly, basically what he found is everywhere he looked, whether he looked at the whole ice-free season, or he looked at monthly chunks of these measurements, he found methane production in the upper layer, in the surface layer, in every system he looked. Sometimes it's very small, but sometimes it can be quite large. Here, these are the various terms, and this is the net difference, net production, expressed as micromoles of methane per liters per day. And as has been found in other research on this topic, the estimates that he got from this mass balance approach, the estimates of oxic methane production, were related to the nutrient status of those lakes, nutrient or chlorophyll. It was slightly more significant for chlorophyll, but basically there seems to be a link to the lake trophic status, which is what has been found in other places. So that's the production side in those small lakes. But now I'd like to turn to the consumption side, which I think is equally, if not more important. And I'll hope I'll convince you of that. And here I call this the importance of getting the oxidation kinetics right. And this is the work of both Shoji and of Paula Reis that have looked at this in multiple different ways. And basically, what they found is that they did a whole bunch of incubations at different temperature, at different methane concentration, and at different oxygen concentration. And these are all incubations that were done at in situ concentration. That is, there was no addition, there was no spiking of methane. This is just done with in situ concentration. And in terms of temperature, they found what is generally found. So there's an increase of methane oxidation with temperature, all else being constant. Same thing, a very strong impact of methane concentration. The more methane you have, the more oxidation you have, typical of a first order reaction. But what they find that was not expected was that instead of this typical increasing function with oxygen, they found a decreasing function. In fact, they kind of found a hump shape. Oxygen being a substrate, you obviously need, unless we're talking about anaerobic methane oxidation, and if we're talking about aerobic methane oxidation, of course, you do need oxygen for that to proceed. And what they found instead of this, which is kind of unusual, thinking of oxygen as a substrate for this reaction, is that the highest oxidation rate occurred when oxygen was actually very low. And at high concentration, it was strongly depressed. And the same thing was observed, the same type of thing was observed looking at the actual cell counts, the cell counts of the methanotrophic bacteria, or methane oxidizing bacteria, known as the mob. And there's two main groups, the alpha mob and the gamma mob, and especially the gamma mob, which are really the more abundant ones. There was this clear negative relationship between oxygen and the abundance of these cells. And the abundance of these cells, much like the incubation experiments here, showed a very strong positive relationship. Interestingly, when bacteria, when in a lake or in a reservoir, there's this sweet spot of high methane concentration and low oxygen concentration, well, those methanotrophic bacteria are really thriving. And it's incredible that they can reach actually very large proportion of the microbial assemblage right at that spot, where that sweet spot that really is the best environment for them. Typically, mobs constitute only about 1% of the total bacterial population. But in that sweet spot, they can go up to 20, and we even had one sample where it got up to 60% of the cells were actually mob cells at that place. And in addition to that, those gamma mobs are really much bigger than heterotrophic bacteria. There's a big difference in size, typically about 3 and more fold difference. Now, this is quite interesting, and I just wanted here to show you a microscopic view of the mob cells and all the heterotrophic bacteria, the DAPI cells, and these are the same field of view. And you can see how much less abundant they are, but much bigger they are, especially the gamma mobs. And they have this incredible ability to process carbon. In fact, if we look at their ability to process carbon, their respective ability to process carbon, on one hand, the methane oxidizing bacteria, on the other hand, the heterotrophic bacteria, there's a big difference. So methanotrophs are capable of using much more efficiently a molecule of methane than the heterotrophic bacteria can of a molecule of dissolved organic carbon, for example. And if we unpack this box plot, we can see that this consumption of carbon by the mob biomass is directly an increasing function up to a plateau of methane concentration itself. So if we look at the oxygen or methane to oxygen molar ratio at different places in the lake, we can measure actually the total consumption of carbon that is funneled through methanotrophy or through heterotrophy. And what was clear is that there is a clear threshold above which the methanotrophs actually process carbon faster than heterotrophic bacteria. And that threshold is about a 0.2 molar ratio. And if we express this even at the whole ecosystem scale throughout, let's say, an entire ice-free season, we estimated that for several lakes, we estimate that portion in several lakes, and we were able to see that in some lakes, more than 50% of the whole bacterial sea consumed is through methanotrophy. And of course, for this to occur, you need to have a thermally stratified condition, because this is what creates the sweet spot that is necessary for them to thrive so much. And this appears to be related to the amount of DOC or the light penetration in those systems. The darker it is, the more of the carbon is funneled through the methanotrophic pathway. It's this kind of this parallel microbial loop, not based on dissolved organic carbon, but instead directly based on methane. And what are the implications of that for methane fluxes to the atmosphere? Where there are several, and one of them is that you can see that very sweet spot here, and the blue is the methane concentration. And what we found in several places we've measured is that there's oftentimes a depth at which the concentration of methane is minimum, lower than what we find at the surface, but precisely where that sweet spot is. And this is directly linked to this nonlinear function with oxygen. Because normally if plenty of oxygen was better for the methanotrophs, well, this is not where what we would find. We would find the minimum more in the surface. And this is basically what we find in many systems. And the red curve here represents the isotopic signature of the methane. And again, strongly indicative of really high oxidation, going from -80 to 0 in terms of isotopic signature of methane. By the way, we found basically the same thing in a tropical reservoir, which I will speak more in detail in a few minutes. And that telltale sign is that we find typically this peak of the delta C13 of methane lower where there is low oxygen. So again, this kind of peak below the thermocline. Here, I should have said that this is only the hypolymian that you're seeing. That's why it's cut off at 10. Well, we see this peak at low concentration of oxygen because high oxygen induces an inhibition of methane oxidation. Now, this is just a little simulation that I've done in a model representing the methane profile. This is on a log scale on the x-axis, and this is depth. And I want you to show that it is strictly this nonlinear curve with oxygen that creates this minimum that we saw during the ice-free season. And this is perhaps more clearly shown here. So this is two profiles, simulated profiles, one expressing this inhibition of oxidation by oxygen in red, and blue without this inhibition, so corresponding to these various curves, except that the colors are mixed. But I want you to notice two things. Knowing this small biogeochemical process induces a very large difference into what you're gonna see in terms of concentration at the surface. This is on a log scale. There's basically more than half of an order of magnitude difference in those two concentrations, given that we have this nonlinear curve. And only with this nonlinear curve do we see this trough deeper in the lower than the thermocline. All right. Now I'm going to move to reservoirs. Reservoirs, there's lots of them. Basically, this is a repertory of the big reservoirs. You know, they have to be included in that database. They have to be higher than 0.1 mega cubic meter, and the sum of those is equivalent to about 350,000 square kilometers, or that is about 10% of all inland waters. Okay. But there's future dams coming in this very important paper by Zarfel in 2014. There's lots of dams that are planned or that are even under construction. So the question is, okay, what are the fluxes at this very large scale from reservoirs? Now, the emission pathways in a lake and in reservoirs are somewhat similar, but they are also different, in particular for hydropower reservoirs, because in addition to diffusive effluxes of both methane and carbon dioxide, also the ebullitive fluxes of methane, in hydropower reservoirs, you have what's called degassing and downstream emission, because water has to go through the turbine, and when it's liberated in the downstream river, a lot of it is degassed and emitted. Now, we've been developing with Sarah this model. It's a series of models that we call the JHA School, and basically it's a series of empirical models that were built from data extracted from the literature. And we build 4 different statistical models, empirical models for the different pathways: diffusive, bubbling, degassing of methane, and as well as CO2 diffusive emission, which we've partitioned into what is sustained by natural processes and what is sustained by the fact that we flooded the landscape, so the anthropogenic part. So I won't go into the details of this. This is a tool that is freely available, and it's been used quite a bit now over the last few years. And basically, these are just some of the empirical models that are embedded in there. So the age of the reservoir is very important. What we see is a very rapid decline of CO2 emissions with age. And of course, the magnitude of emission depends on the local temperature. It depends also on the carbon content of the soil that was flooded. For emission, age of the reservoir was particularly important, and as well as the temperature that you see here. And here's a table of the other factors that are included in here, but I'm not showing as a graph. So when we compare the measured fluxes with the GRES predicted fluxes in terms of different pathways, we get a reasonable agreement. At least there doesn't seem to be any systematic bias. There's a lot of variation, I grant you that, but there's no systematic bias. And the relative proportion of these different pathways vary tremendously from one reservoir to the next. For example, and this is a word from Cynthia Sued, in Borneo, in Batangai Reservoir, the diffusion part, that is what's evaded at the surface, is important, but not so important. In this particular reservoir, in fact, almost 90% of its greenhouse gas footprint is from the downstream emission, both the degassing and what's going on further as you go downstream the river. And I'm just going to very quickly show that, in fact, the methane oxidizing bacteria really take advantage of that, because in that system, the water that goes through the turbine is taken quite deep, where the concentration of methane is really, really high. And as it comes out, it is used by methanotrophic bacteria. And even over a short stretch of about 3 kilometers, already the methanotrophic bacteria have consumed about 15 to 20% of that methane release. Now, this is for only a single system, but how does it fare globally? What are the major pathways of greenhouse gas emissions from reservoirs? So to answer that, what we did is we applied the GRES model to all reservoirs that we could get the necessary information on. The GRES model is unique in that it takes into consideration the very characteristic of every individual reservoirs on which it is applied. And this is work we have published recently with John Harrison. And basically, what it shows here is a map of where gas flux, GHG flux, is occurring in the globe, depending, of course, on where the reservoirs are and their aerial rate. The difference between the upper and the lower panel, the upper panel shows the intensity, so the emissions per meter square per year in all of these different places. And on the right here, you see basically the average of that per latitudinal band. And what basically, before I go there, in the bottom panel, it's more or less the same thing, except that this is the actual magnitude of the flux per 1 degree of latitude. So not per meter square, but the magnitude in terms of gigagrams of carbon dioxide equivalent per degree of latitude per year. And what this shows basically is that the CO2 flux is really the dominant pathway in the boreal region. And as you go down in the subtropical and tropical region, it's really the methane diffusion, the methane degassing and the methane ebullition that takes the biggest chunk of the emission. That is, this is where and the pathway that is most pronounced there. Overall, the overall reservoir GHG fluxes in the tropics and subtropics is where it is highest, and globally, the main the dominant pathway is actually methane degassing, and but note also that is the most uncertain of the pathways that we have, simply because of the model are very uncertain for methane degassing. Now, this provides a snapshot. This is in fact a year 2020 flux at the global scale. But one question that this work doesn't address is, okay, where does that snapshot in time fits in the overall evolution of methane emission from global reservoirs? And we've addressed that by looking at the GRAND database and applying the same GRES model, but through time as the reservoirs are built and put online, so to speak. So this is the cumulative reservoir area around the world from 1900 to 2020. In fact, we go to 2040 by using the assumption that that half of the planned and construction reservoir in the ZARFL database are actually going to exist, and we've kind of distributed them equally between 2020 and 2040. We can discuss that later on. But basically, what we can see is that this is how these two graphs are identical. They're just showing different different aspects of it as to where they are located in tropical and versus subtropical, temperate, and boreal. And what you can see is that past 1980, the contribution or the proportion from boreal reservoir is fairly stable, and where we see an increase is in the other three categories. Same thing, the really, really big reservoirs were mostly built prior to 1990. And from now on, there's some, we can think of the Three Gorge Reservoir in China. There's some that are very large, but in general, the really large one, it's really the mid-sized one that are increasing all through this later period. Now, this is reservoir per se, reservoir surface, but what about emissions throughout that century? And this is what we've been able to ascertain applying the GRES model. And basically, again, these are exactly the same graphs, but colored according to climate, according to pathway, and according to reservoir size class. And the first piece of good news from this graph is that actually the peak of emission is behind us. It occurred in the 80s and 90s. And in those times, not only is the peak then, but the pathway is clearly changing. At that time of the peak, CO2 diffusive fluxes was really the dominant pathway through which GHG was emitted from reservoirs. And as we move along, it's really the degassing and ebullitive flux of methane that is providing the bulk of that of those emissions. Now, that's the first piece of good news, that the peak of emission is at least behind us. But that doesn't mean that the peak of their effects of these emissions is behind us. And this is the kind of last section of my presentation, is we can also look at the impact of the reservoir emissions on climate. But we cannot, even though we have the emissions, we cannot really simply multiply, for example, the methane emissions by their GWPs, because embedded in there, baked in there, there's a time horizon of 100 years or a different number of years, whether depending what you choose. So to really have an effect, to really have an estimate of what is the effect of the reservoir emissions through time, this is really a product of the emission time series with their residence time. It's the convolution really of these two things, because if you put a ton of methane in the atmosphere, of course, it will decrease through time simply because of atmospheric chemistry, in particular reaction with hydroxyl radicals. And in the case of CO2, there's several pools going being removed at different rates, depending if it's oceanic or terrestrial sinks, etc. And you have to multiply these by their respective radiative efficiency of the two gases. So just to give you a quick example on that, for example, if this is the emission curve of a new reservoir that you build, you have to convolute it with its residence time, so-called the adjustment time in the atmosphere. And what you see is this is the effect of those emissions on the atmosphere, because of course, as you move along, yes, the emissions decrease, but the emissions, the past emissions, have themselves a bit disappeared, but not completely, such that the maximum effect is occurring with about a 25 years delay between the emission peak and the peak of the effect. And lastly, I want to translate this, which is what we did, this convolution, to look at how the various, the timeline of the various reservoir-induced radiative forcing is expected to go. And past 2020 here, we have put in shade, kind of bounded by different scenarios of what's going to happen, because the radiative efficiency is itself a function of the mixture of gases that you have. And what I want to show you is basically that as we move on in time, again, it's the ebullitive and the degassing portion of the methane that's going to be exerting the largest radiative forcing. And this is the fraction of the global radiative forcing that is induced by reservoir. So at the peak around 2000, 2%, a bit more than 2% of the total anthropogenic radiative forcing was due to emissions, past and current, from reservoirs. And so this is for the global, but this is for each of the two gases separately. For CO2, it's only about 0.6%, so less than 1%. But it's very interesting to see that for methane, the contribution of reservoirs to the global methane radiative forcing is about 6% around 2000, year 2000. The good news with, if I can call this, that it is the degassing and ablative portion of this, is that this maybe indicate a potential for mitigation. Most of the reservoir-induced emission and radiative forcing are due to this degassing and ablative pathways. Well, perhaps we can do something, both for the ablative, perhaps with a better control on water level, which Beaulieu has shown that it's very important, and also by the position of the turbine intake relative to the methane profile. And my last slide is really an example of this. This is we're back to Batang I, where the dashed line here is where the intake is for that particular reservoir. And this is concentration here. And we've done some simulation that if the intake gates for the turbines had been placed just a few meters above, about 5 meters above to where it is now, it would have reduced the footprint of that reservoir by 85%. So there's some room to maneuver and to better mitigate some of these effects. And with that, I would like to thank you for your attention and also thank ASLO for this invitation. Thank you very much.